Okay, initiation, let's go. So basically what ends up happening to start RNA synthesis is a bunch of stuff have to come together at the promoter to initiate the process. So the promoter is where RNA polymerase, the enzyme, binds. Now, here's the thing about RNA polymerase. It's not just one protein. RNA polymerase is a number of proteins that come together. They stack up together and participate in RNA synthesis. The promoter is obviously called a promoter because it's a sequence that promotes gene expression, and it directs RNA polymerase to go in a specific direction. So basically what you're going to find is your basic gene, like we drew before. Here's your gene. And the promoter is going to be what we call upstream of that, okay? Here's P for promoter. And so as the promoter sequence starts interacting with different elements of RNA polymerase, different chunks of the polymerase, I guess a way to think about it is that an RNA polymerase enzyme, in fact they call it the holoenzyme, is made up of a number of different items. So like this bottle of ink would be one component, this bottle of ink would fit together with it like this is another component, and then brain from Pinky and the Brain would be the part that actually does the reading of the DNA, right? There we go. All right, we're getting wacky today, right? Now, how do these pieces of the RNA polymerase know where to land? Well, within the promoter sequence, there are two regions called consensus sequences. These are short, short, very specific sequences that allow the binding of elements of RNA polymerase. So chunk number one. So let's say, for example, here's my arm, and my arm is the gene, and this region, my hand, is the promoter. One segment would bind at the -30, at the -10 box, and we call this the Pribnow box or the Tata box. Most people call it a Tata box. Haha, very funny, right? Because you have the nucleotides TA and TA. One part of the RNA polymerase sticks there, and it's -10 because it's 10 base pairs above the start site. At -35, you have another little box, another consensus sequence that another element in RNA polymerase binds to. And then a few other things come together. The brain comes here, associates with both of them, and is able to read the DNA sequence to make RNA. So here's brain reading the sequence, okay? And actually doing the work of producing the new strand of RNA from the actual gene. My watch here would be the start, okay? The start site. So you have the -10, -35. You have other elements like this coming down here that would be able to read the DNA strand or read the gene and actually produce the new piece of RNA. I'm going to be posting a couple videos on transcription, initiation, elongation, and termination. So here are our sequences. Most of these are very consistent among almost all genomes. You'll have one at -35, -10. They may be a little bit different between bacteria and eukaryotes, but you're always going to find these consensus sequences upstream of the start. So right here, +1 is where you're going to find A of the ATG. Okay, sorry about that. I heard all of a sudden, Mom! There was no panic that needed to be had. It was funny, though. My daughter put on a big blue wig, and my son didn't know she had a big blue wig, and so he walked in on her wearing a big blue wig and thought she had dyed her hair, and being 13 years old. Anyways, okay, back to consensus sequences. What we're looking at here is a series of regions in different organisms. The lac operon, this is the promoter before the genes that are used to make elements that allow bacteria to consume lactose, for example. Rec-A, these are the elements that are upstream of the gene called Rec-A. Don't worry about what the gene does. You don't need to worry about these names here. What you do need to be aware of, though, is that they're slightly different, but the consensus among all of these regions in all of these bacterial sequences are TTGACA in the -35 region and TATAAT. And so these two regions are essential for the binding, the upstream binding of these elements in RNA polymerase. I guess I should do it this way so it's consistent with what you're reading. I don't know. Okay, so E. coli, being a bacteria, has, again, a holoenzyme. The holoenzyme means you have numerous pieces of protein, each having a different name and coming from different genes themselves, that come together to allow the RNA polymerase enzyme to be constructed and to work. So if you go back and you think about protein structure, this would be the quaternary structure of RNA polymerase. There are 5 subunits in the core. You have 2 alphas, 2 betas, and an omega, and then 1 sigma, okay? So there are 6 units that come together to make the RNA polymerase enzyme in E. coli. And actually that's pretty consistent for a whole lot of different bacteria. Sigma factor itself, this one unit, just sigma alone, is what recognizes -35 and -10. Remember when I said you have these 2 elements that bind to -35, one that binds to -10. I'm trying not to break things here. And then you have one in the middle. This would be sigma. Brain would be the sigma. Pulling these together, allowing them to function properly. And then once RNA polymerase is put together, it opens up the double helix. It breaks the hydrogen bonds between the 2 sides and allows DNA synthesis to occur. And so here's an example of the helices. The helices of RNA polymerase fits within the major groove. Don't worry about alpha helices in the turn right now. Sigma factor is also what starts the synthesis reaction. It's pretty cool. There are 17 base pairs that open up to 17 base pair. Well, the 17 base pairs adjacent to the Tata box in the -10 and -10 region allows these segments to stick. And then initially there's a little small piece of RNA that's produced that allows sigma factor to really attach strongly. I'm not necessarily going to ask about that on the exam, but it's a fun little fact. So here's what happens. Here's the rest of the omega, alphas, and betas, and then here's sigma factor. Sigma factor produces this little kink in the DNA strand, okay? It cruises down here, stabilizes in the promoter region, all right? And then RNA, this short piece of RNA is produced, and then sigma factor, after that short piece of RNA is produced, it actually kicks off sigma factor. When that happens, the RNA polymerase goes down the line. You'll be seeing that in the video that I post. So that is initiation. Elongation is basically where RNA polymerase is saying, okay, on the DNA, I see a C. On the RNA, I'm going to put a G. On the DNA, I see an A. On the RNA, I'm going to put a U. Because remember, you can't put T opposite of A in RNA. All right? And it goes from a 5' to a 3' direction. Ha ha, we're back to the nucleotide. Remember this thing? Carbon, 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 carbon. Here's a base. I'll just put a B for base. Since this is RNA, this is going to have a hydroxyl bound to carbon number 1. There's going to be a bridge here between the carbon, the oxygen, carbon. I'm sorry, that should be a phosphate. Carbon goes here. And the next carbon, oxygen, carbon, carbon, carbon. Here is the 1', 2', 3', 4', carbon 5'. At the very top of this molecule, until modification occurs, you know that this piece of DNA, that this is the top, because you have 3 phosphates here. The bottom will always be where you have a hydroxyl hanging off the 3'. So 5' is top, 3' carbon is the bottom. So 5' carbon, 3' is the bottom. And you always know because you have 3 phosphates here and a hydroxyl here. All right. DNA or RNA polymerase does not do any proofreading, which is problematic because if there's a mutation, if there's an error, you have a problem on your hands. It does form bonds, this phosphodiester bond right here. The phosphodiester bond is the phosphate, the oxygen, and the carbon here, all right? And so this is the sugar phosphate backbone. The sugar is ribose. The ribose sugar phosphate backbone. And here you can actually see the sugar phosphate backbone represented here on the blue RNA strand as a ribbon. So the DNA strand is used as the template for the RNA strand. The opposite DNA is actually called a non-coding strand in most cases, okay? The opposite DNA strand, well, you have the coding strand that has the gene in it, okay? And then you have the non-coding strand, all right? Am I saying that right? I'm actually saying that backwards. My apologies. The RNA strand that's being read from is the non-coding. The opposite is the coding. My bad. Sorry. I always do that, actually. It's funny because if you were in my face-to-face class, I make that mistake every year. I don't know why I say it that way. I know better. So, in addition to that, T is substituted for U, which you guys know, right? T is substituted for U in RNA versus DNA. And genes get transcribed from all over the place. Interestingly, fun fact, even though the promoter is here, all right, so this gene is traveling in this direction. It would be read from left to right. This gene would be read from left to right. This promoter is in this direction. It's being read right to left. I know that this isn't normal for us, but having said that, some genes run right to left or left to right. So now termination. There are two ways in E. coli that termination occurs. Termination can be what we call row dependent or row independent. Row has to do with this protein called the row protein. If the row protein is a part of this organism's metabolism, then the row protein is what kicks off the RNA polymerase. If it's row independent, then you don't require the row protein. So I am going to, hopefully I can find the videos that identify row dependent and independent termination. All right. Now I am having a little bit of, there it is. My apologies. I'm having a little bit of drama with my tech here, as somebody put it. Very well put, by the way. There we go. So what happens here in this particular case is as the RNA is stringing off, as the RNA polymerase is reading and the new strand of RNA is stringing off, there is a small section here called RUT, row utilization site. Row is where the row protein is going to bind. The row protein is kind of cool because what it does is it produces something called a secondary structure. This structure is the stem and loop. So the stem loop structure is actually something that strands of DNA and RNA do not like. What ends up happening is hydrogen bonds can form there if you have like a C and a G. And what it does is it puts pressure on the strand and the RNA polymerase, allowing at the termination point here, allowing release of the new RNA strand from the RNA polymerase enzyme. Once that happens, row factor is released, boom, it's gone. And so in this particular case, what row is doing is it's racing along and allowing this loop and stem to form. Once it catches up with the RNA polymerase, it kind of gums up the works and allows things to release. Interestingly, over here on the termination end, you have a whole lot of A's. It's called a poly A tail. You can have an additional poly A tail put there, but if you have lots of A's, the number of bonds between A's and U's, or A's and T's, are 2 hydrogen bonds, which means it's easy to break. All right, so we're going to talk about the next step in RNA synthesis in the next